Tom grew up in New York. As a matter of fact, as a child, he bicycled down to the site of the George Washington Bridge as they were building it. Tom became interested in science because of the Depression. He really wanted to be an English lit teacher. He went to Princeton, and his father said to him, "You will have to get a job because it's the middle of the Depression, and you're not going to get it with English literature." So he chose chemistry, and that's what led him to being interested in science. After he got out of school, he went into industry, and the first thing was that they called him into the office and said, "Here's a vial of a numbered molecule, and we want you to develop this for the market." And it's a carbonic anhydrase inhibitor. And carbonic anhydrase is an enzyme. And he always said that he took the vial and he went into his office and locked the door and looked up enzymes and carbonic anhydrase because he had never heard of either one. That was his introduction to a passion.
He was very widely known for his work with carbonic anhydrase. Dr. Maren really originated the field. He was present right at the start on the medical implications of the inhibition of carbonic anhydrase. Carbonic anhydrase is an enzyme that's in many, many cells of our body. It's involved in many physiological processes, in respiration, in fluid secretion, in the function of the kidneys. In the area of fluid secretion, it's involved in the formation of ocular fluid, hence the relevance to glaucoma.
Tom's focus was on studying the permeability of different carbonic anhydrase inhibitors so that he could get a topical drug to work for glaucoma. He was studying compound after compound, trying to define in the eye what controls the permeability, what controls allowing a drug to get into the eye.
For decades, researchers thought that it would not be possible to have an eye drop that would be effective inhibiting carbonic anhydrase and controlling glaucoma. Tom was a fighter, and he believed that this could be done. And that was his genius, that he was able in the lab to produce carbonic anhydrase inhibitors that would pass across the cornea and be useful in the control of glaucoma. Tom needed, of course, a pharmaceutical company to get the drug out onto the market. When Merck saw that this could be done, Merck took up the project. They tweaked Tom's discovery to get a medication that perhaps would pass the cornea a little more efficiently, that would still inhibit carbonic anhydrase.
And lo and behold, here comes Trusopt. He and the school licensed Trusopt to Merck. And for several years thereafter, it was a leading, if not the leading, source of royalty money for this university. He's an example of a methodical, persistent approach to innovation. I mean, it wasn't like he woke up in the middle of the night and suddenly say, "Oh, that's how it works," or that's what. I mean, he's been in love with carbonic anhydrase since the middle of the last century and its inhibitors. And he studied this in all sorts of creatures and all sorts of organs of the body.
Tom would be very pleased to know that carbonic anhydrase research exists at the University of Florida, is ongoing and making new discoveries and more progress.
Tom's thoughts about being inducted into the Hall of Fame would have been good. He was proud of what he had done, and I think he would be proud of this.